Nikolas Van Nelson, and my presentation is discovery of protein Affibodies for cancer detection and treatment. So a brief introduction to my project. TROP2, which is a receptor on cells, was chosen as a target because it is overexpressed on tumor cells. This makes it a good target for treatment and diagnosis because it allows preferential targeting of the cancer cells, as seen on the figure on the right. Proteins that bind to TROP2 can either deliver a cytotoxic payload for treatment or a radioactive tracer for imaging. Due to how many variants of proteins there needs to be a method to test large quantities of proteins at once, which is done with a yeast surface display. I started the project with an Affibody library that had a diversity of 543,000 after the 3 rounds of magnetic activated cell sorting, or MAX, which was done by Hammer, a graduate student from my lab. The Affibody library is made of proteins with 58 amino acids that form 3 helix bundles. The project was started with a 1 to 5 nanomolar TROP2 binding affinity, fluorescent activated cell sorting, or FACS, which was then immediately followed by a more stringent sort by using a 10x diluted concentration of TROP2 receptors. This sort was done due to excessive noise from the first sort, which will be seen in later slides. The binding affinity FACS sort is depicted on the diagram on the left, where the protein on the yeast surface display are binding to TROP2 receptors from lysed mammalian cells. This complex was then treated with anti-c-Myc FITC and anti-biotin Alexaflora 647 antibodies. Yeast cells that had both signals were considered binders and sorted based on how strong the Alexaflora 647 signal was. If there is no Alexaflora 647 signal, it means that the protein is not a binder, as now depicted on the left, and the yeast surface display was then not collected. The next step was a stability sort, where the yeast surface display was treated with protease K, depicted on the right. More stable proteins were able to stay intact during the treatment, so the yeast surface display was able to have the anti-HA Alexaflor 647 and anti-c-Myc FITC antibodies bind to the HA and c-Myc terminals. If the proteins were not stable, they were cut, preventing the anti-c-Myc FITC antibody from binding to the display, as now shown on the right. This makes any proteins that were cut not emit the FITC signal. The displays are then sorted based on how strong of a FITC signal for each yeast surface display. The yeast populations after each sort, including the population after the 3rd round of MAX, will be sent for deep sequencing to then compare enrichment of clones after each sort, which will allow for the selection of top clones for further testing. For the first sort, which was a 1 to 5 nanomolar TROP2 affinity FACS sort, this used the population after the 3rd round of MAX, resulting in the P5 and P4 populations, which include the top 0.1% and the next best 0.1% of binders, respectively. The P5 and P4 populations were combined for the next sort, resulting in a combined diversity of 22,000. There's a lot of noise, as seen by there being yeast with high Alexaflora 647 signal and low FITC signal on the bottom of the graph. Using that combined P4 and P5 populations from the previous sort, a more stringent sort was done with a 10x diluted concentration of TROP2. This resulted in a new P5 and P4 populations, which include the top 0.4 and the next best 0.5% of binders, respectively. There's significantly less noise in this sort, as seen by there being less yeast with low FITC and high Alexaflor 647. And comparing the negative control to the sample, there's a substantial increase in signal for yeast, where the proteins bind to TROP2. The resulting diversities of P5 is around 6,000 and P4 is around 7,000. Next was the stability FACS sort of the P5 population. This was sorted into 4 gates based on stability, with P5 being the top 10%, P4 being the next best 15%, P3 being the next best 25%, and P2 being the remaining 50%. The red yeast on the graph did not have much Alexaflor 647 signal, indicating the proteins were not expressing, so they were not collected during this sort. The P4 population was gated similarly to the P5 population for the stability sort, but there's slightly less of a gradient in the FITC signal. Moving on to the next steps. First, I'll be selecting the top clones by deep sequencing the yeast populations after every fact sort, including the initial population, and then look for enrichment of DNA sequences of clones to select top performing clones. I will then move on to analyzing the top clones. This will be done by producing the proteins in E. coli, then characterizing them by measuring solubility expression yield, which is how well they can be produced, finding binding affinity for TROP2 present on mammalian cells, and get the melting point, which is a measure of stability through circular dichromism. Thank you for listening to my presentation.